Every cell in your body is constantly making a life or death decision, and for the MCAT, you need to know exactly how that choice is controlled. What actually triggers a cell to self-destruct? And how do signaling pathways like intrinsic and extrinsic apoptosis show up in passage questions? In this video, we'll break down the key molecules, pathways, and high-yield connections so you can confidently tackle any question on cell signaling and programmed cell death. Apoptosis is a tightly regulated form of programmed cell death, characterized by specific morphological and biochemical changes. Unlike necrosis, which is messy and inflammatory, apoptosis is clean and controlled, making it essential for development and tissue homeostasis. During apoptosis, cells shrink, chromatin condenses, DNA is fragmented, and membrane blebbing occurs, eventually forming apoptotic bodies that are phagocytosed without triggering inflammation. A key biochemical hallmark is the activation of Caspases, a family of proteases that dismantle the cell from within. On the MCAT, you should be able to distinguish apoptosis from necrosis, especially in passages that describe inflammation or lack thereof. A helpful mnemonic is apoptosis is A+ orderly, meaning it's organized and beneficial. Real-world examples include the removal of webbing between embryonic fingers and the elimination of damaged or potentially cancerous cells. Apoptosis is also linked to diseases. Too little apoptosis can contribute to cancer, while too much can contribute to neurodegenerative disorders like Alzheimer's. Understanding apoptosis also connects to immune system function, as cytotoxic T cells induce apoptosis in infected cells. MCAT questions often test your ability to identify when apoptosis is occurring based on clues like DNA laddering or caspase activation, so recognizing these key features is critical. The intrinsic and extrinsic pathways are the two main mechanisms that initiate apoptosis. The intrinsic pathway, also known as the mitochondrial pathway, is triggered by internal stress signals such as DNA damage, oxidative stress, or lack of growth factors. This pathway involves the release of cytochrome C from the mitochondria, which then binds to APAF1 and forms the apoptosome, ultimately activating Caspase-9 and downstream executioner Caspases. The extrinsic pathway, on the other hand, is initiated by external signals when death ligands, such as Fas ligand or TNF-alpha, bind to their corresponding death receptors on the cell surface. This activates Caspase-8 and leads to the same execution phase. A helpful way to remember this is intrinsic equals internal mitochondria, extrinsic equals external receptors. On the MCAT, questions often ask you to identify which pathway is being activated based on the molecules involved, such as cytochrome C for intrinsic or Fas receptor for extrinsic. These pathways are also interconnected, as Caspase-8 can amplify the intrinsic pathway by influencing mitochondrial permeability. Understanding these mechanisms is crucial for interpreting experimental data and passage-based questions involving cell death. Caspases are central executioners of apoptosis that exist as inactive precursors and become activated in a cascade. These enzymes are cysteine-aspartic proteases, meaning they cleave proteins at specific aspartate residues. They are synthesized as inactive zymogens, or procaspases, and require cleavage to become active. Once activated, initiator caspases like Caspase-8 or Caspase-9 activate executioner caspases such as Caspase-3, -6, and -7. This cascade amplifies the apoptotic signal, ensuring that once the process begins, it proceeds irreversibly. Caspases target key structural and regulatory proteins, leading to DNA fragmentation, cytoskeletal breakdown, and membrane changes. A mnemonic is Caspases cause cell collapse. On the MCAT, you may see experimental setups measuring caspase activity or using caspase inhibitors to block apoptosis. You should understand that blocking Caspases prevents apoptosis even if upstream signals are present. This concept also ties into broader enzyme regulation topics, including zymogen activation, which is also seen in digestive enzymes like trypsin. Recognizing Caspases as the point of no return in apoptosis is a high-yield takeaway. Cell signaling pathways rely on ligands, receptors, and second messengers to transmit and amplify signals within the cell. A signaling pathway typically begins when a ligand, such as a hormone or growth factor, binds to a receptor, which can be located on the cell membrane or inside the cell. Membrane receptors include G-protein-coupled receptors and receptor tyrosine kinases, both of which are heavily tested on the MCAT. Once activated, these receptors initiate intracellular signaling cascades involving second messengers like CAMP, IP3, DAG, and calcium ions. These molecules amplify the signal, allowing a small number of ligands to produce a large cellular response. A useful mnemonic is ligand binds, signal finds, response amplifies. On the MCAT, you should be able to trace a signaling pathway and predict outcomes, such as changes in gene expression or enzyme activity. This concept also connects to pharmacology, where drugs can act as agonists or antagonists of receptors. Understanding how signals are transmitted and amplified helps you interpret experimental data, especially when inhibitors or mutations are introduced into the pathway. Dysregulation of apoptosis and cell signaling pathways is a major contributor to diseases such as cancer. In healthy cells, signaling pathways carefully balance cell proliferation and cell death. But when this balance is disrupted, uncontrolled growth can occur. For example, mutations in genes that promote apoptosis, such as p53, can prevent damaged cells from undergoing programmed cell death, allowing them to survive and proliferate. Similarly, overactivation of growth-promoting pathways, such as those involving receptor tyrosine kinases, can lead to excessive cell division. A helpful way to think about this is that cancer cells ignore stop signals and amplify go signals. On the MCAT, you may encounter questions involving oncogenes and tumor suppressor genes, where you must determine how mutations affect signaling pathways and apoptosis. This topic also ties into treatments like chemotherapy and targeted therapies, which often aim to restore apoptosis or inhibit signaling pathways. Understanding this connection helps you integrate knowledge across cell biology, genetics, and biochemistry, making it a high-yield area for passage-based questions.